get to the specific Griffith's experiment as we go through the experiments, I think it's really important to start by highlighting, why was DNA needed to be proven as the genetic material, and why was protein a runner-up. And the reason for this is that when you think about it, like the code that makes protein strands and protein structure has 20 amino acids, so 20 letters in the alphabet, whereas the code that makes up DNA has 4 letters, right? 4 nitrogenous bases. And when you think about the complexity of structure, that would maybe like create a complex language or complex code that could then carry large amount of information, then you're thinking of like structure, which could be kind of considered like grammar in terms of language, right? And protein has very complex structure beyond just the primary strand. It has folding and secondary, tertiary, quaternary structure, right? Whereas DNA is a double helix that gets wrapped around, which we will talk about later. But that's why protein was definitely considered an option to carry the genetic information. But as we now know, that DNA actually carries the genetic information, and we can kind of look through these experiments to see how that was proven to be. So again, the purpose of these experiments was to identify what the genetic material was, and as you try to understand and grasp each experiment, you should focus on 3 things. You should focus on what was the purpose of doing the experiment, what were the procedures, and what was the outcome. So let's talk about Griffith's experiment, right? What was Griffith's purpose? Well, Griffith was a doctor, and he was dealing with patients, and he found that some of his patients that were exposed to the bacteria were dying, and some of them were fine and lived on. So he performed this set of experiments to kind of figure out why his patients were dying and why some weren't. So there's 2 different types of bacteria. There's the S strain and the R strain. So the S strain is a smooth strain, and this is called the smooth strain because there was a capsule that kind of contained the bacteria within it, and this capsule prevented the mouse's immune system from detecting this bacteria and then invoking an immune response to kill the bacteria. Whereas then you have the rough bacteria, which didn't have any capsule on it, so like you can think of all the membrane proteins kind of being exposed, so it was kind of rough. So this allowed for the mouse's immune system to detect the bacteria and then kill it. So when the mouse was injected with smooth bacteria, its immune system couldn't detect the bacteria, so the mouse died. Whereas when the mouse was injected with R strain, the mouse's immune system could detect it, so the mouse lives, right? And then to further run experiments, Griffith decided to heat kill the S strain, right? And he found that the mouse lived because the immune system didn't need to kill the bacteria was already dead. But when he found that he injected a living R strain and a dead S strain, the mouse still died, and this proves, or this shows, that there is bacterial transformation, because what happened was the DNA, which was still present in the dead S cells, actually made its way into the R cells and then made those R cells smooth cells. So those R cells could now form a capsule around themselves because they have the genes or the DNA to code for that capsule or that feature or that phenotype, right? So now the mouse does die. So the conclusion is that a bacterial transformation in this case, right? So the next set of experiments was run by Avery, and this was to understand which molecule in that S bacteria was being transferred from the S bacteria to the R bacteria to make the R bacteria an S bacteria, right? So was it protein, was it RNA, was it DNA, right? So he made these solutions, and he removed one of each component that he predicted would be the genetic material to find what the genetic material is, right? And if the genetic material of the S bacteria would be present, then you would have a dead mouse and you would not have any S cells appeared. Whereas if that genetic material was there, then you would have a dead mouse and you would have that S cell appear, right? So when he did it with no protein, he still found that S cells appeared. When he did it with no RNA, he still found that S cells appeared. And finally, when he did it with no DNA, he found that only R cells were there and there were no S cells, which proves that DNA was the genetic material that was being transferred. So then we have Shargoff's experiment, and Shargoff's experiment was really to identify what structure DNA had. So he found that DNA was made up of deoxyribose, which is our sugar right here, a phosphate group, and a nucleotide base. And we know that our nucleotide bases are cytosine, thymine, uracil, adenine, and guanine. And uracil is unique to RNA, and then these four are found in DNA, right? So he found out that the way these were paired together so that there was an equal ratio of A and T and C and G, and if that ratio is continuously found in multiple samples of DNA, then it can be concluded that these were in fact paired up in a double helix, right? So that was Shargoff's experiment. And then Hershey and Chase's experiment was again the purpose was to identify DNA as the genetic material. So what they did is they used they used radioactive isotopes to label the different components of a virus, right? So these are little virus dudes. I think they look kind of cool, like little aliens, right? And they labeled the proteins with a one radioactive isotope, and they labeled the DNA inside the head with another radioactive isotope, and they let the bacteria infect these bacteria cells, and then they let them separate out. And what they would do is they would centrifuge it to separate whatever was outside in the extracellular fluid and what was inside the cell, right? So then in this case, the DNA is injected in, right? So all of the radioactive material, which is the protein, is actually left in the supernatant once centrifuged. So they saw the radioactivity in the supernatant. Whereas here, when they labeled the DNA with the radioactive isotope, they found that that radioactivity in the pellet, which was indicative of the radioactivity in the DNA that was actually injected into the bacteria cell. So this indicated that the bacteria cell had DNA and that this was the material that was being transferred over, and that was the genetic material. So this, the conclusion from this is that DNA is the genetic material and that protein is not the genetic material. So these are the series of experiments, the 4 experiments that were done to prove DNA as a genetic material and kind of establish DNA's structure. It's important to understand them. If you have any questions or any clarifications needed, I know sometimes I'm stumbling upon my words. Forgive me, I'm still trying to get used to recording these videos efficiently, but ask me questions please in the comments below, sign in, and I will see you in the next video when we start talking about DNA's structure.